I welcome you all in the online refresher course in the chemistry hosted by Department of Chemistry, Gujarat University, Ahmedabad. On behalf of Dr. Jagdish Joshi, Professor Director, UGC HRDC, and Dr. Dilip Vasawa, Assistant Professor, Refresher Course Coordinator, Department of Chemistry, Gujarat University.
I welcome you all in the session of Dr. Naval Kapuriya, Associate Professor, Member of Executive Council, Syndicate, Department of Chemistry and Forensic Science, Bhakt Kavi Narsi Mehta, University, Junagadh, Gujarat. He has done postdoctoral research in medical chemistry 2012, the Ohio State University, Columbus, USA. He has qualified Gujarat State Eligible Test Chemical Science held in August 2017. He has rich experience in academics as well as in a research field. His contribution in research includes he has published 29 research articles in international journals. His impact factor range is 0.75 to 6.6. 4 proceeding in an American Association from the Cancer Research, 2 books published with a subject anti-cancer agent in an international publication 2018. Here are some glimpses of achievements and awards. Doctoral Fellowship by Institution of Biomedical Science, Academic Sinica, Taiwan. Postdoctoral Fellowship by Ohio State University, USA. Research person by Gujarat State Biotechnology Mission for Entrance Test, Gandhinagar. Discovered a novel rearrangement which provide direct synthesis of aldoxamines from amines. Delivered 5 invited talks in national and international seminars and workshops and conferences at the colleges, states, and universities. Also delivered a talk as a keynote speaker in a national seminar organized by Subhash Academy, Junagadh, 2019. He has membership and affiliation in a professional associations with many national and international collaborators and a reviewer. Visiting lecturer, nodal officer, member of Board of Studies, Board of Examination, syndicate body, resource person, and many more. His broad area of research includes target-based anti-cancer drug design and synthesis, DNA analytic agents as an anti-tumor agent, modification of natural molecules, developing new synthetic protocols and a scale-up synthesis. In addition to that, he has experience in handling sophisticated instruments like NMR, IR, HPLC, and many more. Thank you very much.
Hello everyone, myself Dr. Naval Kapuriya, working as a Associate Professor at Department of Chemistry and Forensic Science, Bhakt Kavi Narsi Mehta University, Junagadh. First of all, I would like to welcome all the participants who have joined this online refresher course in chemistry organized by UGC HRDC Center, Gujarat University. I also sincerely thanks to the Director of the HRDC Center, Professor Dr. Jagdish Joshi sir, and the coordinator of this refresher course, Dr. Dilip Vasawa, for giving me this opportunity to interact with my fellow colleagues. Friends, today I am going to talk on the chemistry of anti-cancer agents, past, present, and future. So, in my presentation, I am going to share 3 important aspects. First of all, we will discuss a little bit introduction about the medicinal chemistry. Then we will move on the cancer introduction, its hallmarks, and we will discuss on the classification of various anti-cancer agents based on the targets. Then I will present some case studies of DNA interacting agents, enzyme inhibitors, so that the new beginners or who want to work in the medicinal chemistry, especially in the anti-cancer field, they can have an idea that how can I start the anti-cancer drug discovery program or process or projects. We'll also discuss the biosimilar, which is the future of the drug discovery, and we'll also talk the chemopreventive agents. So overall, you can say that I will cover from the very basic fundamental medicinal part to the drug discovery or development part, especially to anti-cancer. Friends, medicinal chemistry is not a new word. Actually, if I want to define, it is nothing but the journey of a molecule, right? So the role of the medicinal chemist is to discover and develop a molecule to address the health issues of the society. The medicinal chemistry basically covers three important area or aspects: discovery, optimization, and development aspects. In discovery part, the identification of therapeutic targets for particular disease, for example enzymes or transporter protein or cellular signalings or in vivo models. And second part includes the identification of new substances, either naturally driven or synthetic molecules. We call them as a lead compound, which can interact with the selected target for particular disease. So there is a very important basic part where the biological application of molecule starts. So it is called the discovery part. The optimization parts where the molecule which we have identified for particular target has to optimize to get optimum potency, best selectivity, and less toxicity. For that, the chemist has to work and it has to optimize the structure by manipulating with the functional group or the core unit or doing the bioisosterism. There are a number of techniques where molecule can be optimized. By doing that, a number of series of, or you can say a library of molecule will be generated. So we'll have a structure activity relationship of the molecule for particular target or disease, and by that we can also understand the molecular action or molecular mechanism, that how particular molecule is interacting with the particular targets or a particular action of mechanism. So this is called the optimization processes. Now third important part is development processes. So once we have an optimized structure in our hand, the lead compound in our hand, it further goes for the optimization for the improvement of the pharmacokinetic properties. These pharmacokinetic properties includes ADME, right? So it's administration, or you can say absorption, distribution, metabolism, and excretion part or toxicity part also included. Further, the fine tuning of the pharmaceutical property also needs to be done through proper chemical formulation so that it can be useful for the clinical application. So a molecule has a journey from discovery to development, so nothing but the from lab to clinical application. So this journey of molecule is described, or you can say prescribed, by medicinal chemistry. Fine. Today we are going to talk on the cancer. So before we go and talk on the anti-cancer drug discovery, and we just elaborate on the different aspect of the anti-cancer drug discovery, it is very much important for us to understand what is the disease itself, right? Without understanding the disease, it is very difficult to target the disease by any molecule, right? So the detailed mechanism, the detailing of disease is required. So cancer, friends, we know that it's the most challenging disease, right? And it's not a new disease, in fact, right? It's known since thousands of years, right? And right now, according to WHO, 1 out of 3 person is affected to cancer worldwide, right? And in India, the last 2 or 3 decades, if you have noticed, the numbers are rising, and it substantially increased due to the heavy industrialization that is happening in the India. Also, if I talk particularly in Gujarat, that In Gujarat, the oral cancer is a major problem, and this is nothing but due to the heavy tobacco consumption. Especially our youth are facing this problem very much, and we need to solve this. That's why I said that cancer is going to be a most challenging disease for us, especially developing countries. As I said, it's rising in India, right? Cancer is rising in India. If you look at I'm presenting the data of the Indian Council of Medical Research, ICMR. If you compare the data of 2012 and 2018, right, you can see that the number of cases are rising. More than 1.15 million new cases they have been recorded in 2018 itself. So it's increasing by 15%, just 5 or 6 years. You look at these highlighted marks I have shown here, that lip and oral cavity cancers. This number of cases, you see this, is rising about 114%, right, in just 5 to 6 years. This is alarming situation, as I said in earlier. This is because of the heavy tobacco consumption that is happening in all over India and especially in Gujarat. So it's a high time that we understand this disease, and also we try to focus on anti-cancer drug discovery, so that in future we'll have some molecular drug in our hand so we can treat this disease. Fine. So as I said that, let's go a little bit into the deep and try to understand what is cancer. Cancer, if I want to define or if I want to write a line, is an uncontrolled growth of the cells. Friends, we know that our body is made up of trillions of cells. These cells are growing by microcontrol system of our body. So the cell growth and their multiplication controls are inherent in our body, right? But when this cell growth become uncontrolled, then we call them as a cancer cell. So there is a difference between normal cell and cancer cell. We'll go into little bit into the deep later on, but formally if I want to classify or if I want to differentiate the cancer, there are two types of cancer. Number one is benign cancer. When a tumor or a cancer is localized in one part of the body, then it is called a benign tumor. Generally, benign tumor are not life-threatening. Sometimes if we do the simple surgery, we can remove it. But on the other hand, the tumor which are invading, that means if one part of the body is cancerous and if it start to spread other part of the body, it is called the invading processes, or you can call them as a metastasis processes, and it start to spread all over the body, then we call them as a malignant tumor. This malignant tumor is the most life-threatening problem, right? So we have to understand the difference between these two and how this malignant tumor works in our body and how our different function of the cells are malfunction during the processes that we are going to understand in little bit detail. So overall, if I want to tell you the different types of cancer, I am not telling the exact detail, but there are 4 different types of cancer. Carcinoma is a term which is usually given to a cancer for particular organs or glands. For example, skin, lungs, breast, pancreas, stomach, etc. Okay, which are generally making the lines of the organs. The other type of cancer is sarcoma. Sarcoma is a cancer of connective tissues, for example, bones and muscles and cartilage and blood vessels. These are connective tissues. So the cancer related to these kind of tissues is known as a sarcoma. The third type of cancer is leukemia, where it generally referred as a blood cancer, right? So in other words, it is also known as a bone marrow, because generally blood cells are formed by the bone marrow. So it's a bone marrow cancer. Then fourth type of cancer is lymphoma, which is usually referred to the cancer of immune system, especially lymphocytes cancer. So basically 4 type of cancers. You can classify them: carcinoma, sarcoma, leukemia and lymphoma. Fine, let us go a little bit into the deep. As I said in the beginning, that cancer is an uncontrolled cell growth. That means it has something to do with the cell, right? So uncontrolled cell growth, what is the meaning of this? Fine, so we know that every cell grows through a cell cycle, and that cell cycle has a different stages, right? So G1 stage, S stage, G2 stage and M stage. So in G1 cell, generally cell grows, right? And in S phase, generally replication of DNA occurs. So after replication, then cell prepare to divide. So in that case, the last stage is cell division. We call them as a mitosis, where two cells will separated and there will be a microtubule which will slowly dissolve, and these two cells will be separated. So this is the simple explanation of how cell divide, right? Cell cycle occurs. Now, who is controlling the cell cycle? As I said that cancer is a problem of cell division, uncontrolled cell division. So who is controlling the cell division? So basically the DNA, we call them as a gene, right? Gene which is located into the center of the cell, we call them as a gene or DNA. DNA is responsible for the cell cycle or cell division. So that means if there is a problem in gene, then and then the cell cycle will go wrong, and that's why it will become uncontrollable, right? So cancer is a genetic defects, right? So in other words, uncontrolled cell growth means genetic defects, fine. So let's go a little bit further and we try to understand now how these genetic defects will occur and what is the genetic defects. See this, that the genes or the DNA are located into the cell nucleus, right? So now we are entering into the cell nucleus. The cell nucleus has a DNA and wonderfully arranged through only 4 heterocycles. You know that DNA are made up of 4 heterocycles, that is adenine, guanine, cytosine and thymine. These 4 heterocycles are wonderfully arranged through hydrogen bonding, and they are making a base pairing. We know this is a base pairing. So adenine usually make a base pair with thymine, and guanine will make a base pair with cytosine, right? This is a natural base pairing. So when DNA replication occurs, or you can say the translation occurs, when DNA divide, generally this base pairing is always maintained in natural processes. So then during the replication, when there is a father DNA and this is a daughter DNA, or you can say the new DNA form, you will see that A has a base pairing with T and G has a base pairing with C. So it will maintain in the daughter DNA also. But if some problem occurs and somehow this base pair is mismatched, then what will happen is the new DNA which is formed is a faulty DNA, right? So this faulty DNA will lead to another faulty DNA, because when this cell containing this DNA, faulty DNA, when it will replicate, it will again produce a faulty DNA. Now faulty DNA, or you can say the mismatched DNA, or you can say the mutated DNA, when it is in the center of the cell, it will completely sending the different signals than the normal signaling. So the normal signaling and the mutated DNA signalings of the cells are different. So these slowly become uncontrollable because the DNA is faulty. So the boss, which is the center, the DNA gene itself is a faulty. So whatever message or whatever the regulation or whatever control this DNA has, it is now not up to the level of the normal cell. So this faulty DNA with mutation of base pairing it will produce a faulty cell. So slowly this cell will, instead of stopping the cell division, it will continue the cell division, right? Because the DNA is not original, or you can say the DNA is mutated, right? So it will not listen to this, to the sensory or the regulatory mechanism of our body. That's why you can say that cancer arises when you have a mutation in the DNA. Now one very important question is, how many mutation can lead to the cancer cell? So from normal cell to the cancer cell is a mutation process. Generally, it's not that just one mutation and we'll have a cancer cell. No. In our body, there is a wonderful mechanism which is called the DNA repairing mechanism. So if there is a DNA which is faulty, our body itself can repair the DNA, or if it is not repaired, there is a third mechanism is called apoptosis mechanism, where the cell has to die. The faulty DNA containing cell has to commit the suicide. See the beautiful control, right, of the mechanism. So ultimately the faulty cell will die itself. But when the mutation is so much that the mechanism is stop working, right? So first mutation, second mutation, third mutation, or you can say the successive mutation, this is happening over the age. Generally after 45 or 50 years of age, when our immune system is down, then generally the faulty DNA will survive, right? When faulty DNA will survive, so the mutation will survive, the successive mutation will survive. So these successive mutations will ultimately lead to a malignant cell, which is unwanted cell for our body, and this will continuously divide. So what will happen is, a particular part of the body or organ, you will find a unwanted cell, we call them as a tumor, right? So the genetic defects which make faulty DNA, which lead to the abnormal behavior of the cell, because this is abnormal behavior because of due to the mismatch pair or faulty DNA. Fine. So what we think, what we learn that cancer is a problem of uncontrolled cell growth. Then we understand that this uncontrolled cell growth happening during the cell cycle. Now the cell cycle is controlled by the DNA, so there is a problem with the DNA. That means the DNA has some mutations. Mutation in the form of the chemical alteration. Chemical alteration can be number of types. It's not just a mismatch of base pair. Sometimes you will see alkylation, sometimes you will see oxidation, sometimes you will see a number of modification on DNA, because DNA is, as I said, is nothing but the 4 heterocycles. So if there is a change in the structure of the DNA, it is called the mutation. So that mutation will lead to the abnormal cell behavior, and that's where cancer arises. Fine. Let us go further. The most threatening problem of the cancer is its property of tissue invasion and metastasis. See that the normal cell, when they are produced, they have a E-gathering protein, an extracellular matrix through which the normal cell will adhere to the surface. So the normal cell will not migrate from one part of the body to another part of the body, because they will adhere to the surface. They will stick to the surface of the particular tissue or organ. But in case of cancer cell, they do not have this adhering protein. Also, so they will easily break away. And for special things that is with cancer is, they have a matrix metalloproteinase. They have a MMPs, matrix metalloproteinase. This is a enzyme which is hydrolyzing the protein barrier, and that's how it will start to migrate from one tissue or one organ through the blood vessel to the another organ. So that's why sometime if you remove a tumor from one site or one gland or one organ, you don't know that it's already started to growing into the another part of the body. So this process is known as a metastasis process. In layman languages or layman process, generally what doctor says that it's a last stage cancer, or it is always said that third stage or fourth stage cancer. When there is a third stage or fourth stage cancer, that means it's already in the invasion process. It's already in the metastasis process. This we need to understand. Fine. So when you start to invasion, where it will go? So usually when people have a breast cancer, the main metastasis sites are lung cancer, liver cancer, or bone cancer. So here I have given some list where different type of cancer and their metastasis site. So often you will find that people have a colon cancer, they will also have a, after some days or some time, they will find a liver cancer or lung cancer or peritoneum cancer. Similarly, if a woman has a ovarian cancer, you will see other metastasis site cancer later on. If it has a surgery, doesn't matter. If it is in the metastasis process, it will start the new cancer at different site. So friends, you can understand that cancer is not a simple disease. It's a very complex disease, and because it's made up of our body, the cell of the cancer is made up by our body itself. That's why it's not giving any symptoms to us, right? So it's a normal process. Division of the cell is a normal process. That's why we are not getting any abnormal signalings from our body until and unless we have a tumor at particular organ or glands. So metastasis is a process through which the cancer spreads across the body. Fine. So far we talked about that how cancer is starting in our body, what is cancer, and how it invades. So if you compare the normal cell and cancer cell, these are the hallmarks of the cancer cell. The first and foremost hallmarks of the cancer cell that it has a abnormal signaling pathways. It does not obey the normal signaling of the cells. Similarly, it has an insensitivity to growth inhibitory signals. See that once we have a desired number of cells, requisite number of cells in hand, naturally the microcontrol system of our body will inhibit the process so that no more cell will grow. But when we have a cancer cell, it do not obey the rule, right? So it is insensitive to growth. Similarly, as I say, it has abnormal in cell cycle regulation. In cell cycle also there are number of ways you can control the cell cycle. There are number of molecules, there are number of checkposts are there through which the cell cycle progress. But cancer cell cycles always, once it start, it never stop, right? That's why we'll have unwanted number of cells present in our body. Similarly, as I said, the apoptosis mechanism, evasion of programmed cell death. Sometimes when body has a faulty DNA, body can order, our control system can order the faulty DNA or faulty cell to die. This is called suicidal mechanism. But in case of cancer, this mechanism is not working. That's why the faulty DNA cell will survive. And as I say, it will start the limitless cell division. It is a no stopping, right? So tissue invasion and metastasis will occur. So these are the hallmarks of the cancer, which is make a clear difference between the normal cell and cancer cell. So now after understanding the cancer, now let us try to understand how to detect the cancer. So there are basically three types of detecting techniques right now in the use, or you can say in the format of the prescription. Number one is mammograms, where we can take an X-ray picture of the organ to see that particular over mass, or you can say the over cell are present or not. Another very frequently used technique is CT scan, which is called as a computed tomography. So this is a CT scan. So CT scan, what generally they are doing the cross section analysis of the body, of the particular organs or bones or other tissue, and they can find out whether there is unwanted mass is present or not. And third type of technique is, now it's a laboratory test, and you often heard this name, that is a biopsy or the pap test, especially for uterus or cervical cancer, and the tumor marker. So sometimes when cancer grows, it produces some chemicals, unwanted chemicals in our body. So if we can detect that that markers, then also we can detect whether there is particular cancer is growing or not. So these are the different type of general idea I am giving you, the different type of screening test for cancer. So we learn about the cancer. It's a problem of faulty DNA, successive mutation, and then there are the different hallmarks of the cancer. And we say that once there is a cancer produced, how we can detect. So there are three different techniques through which we can detect. Now let us talk about the treatment option for cancer. So before having a treatment option, right now there are 5 different modalities or 5 types of different facilities, I can say, available for the treatment of cancer. The number one is surgery, radiotherapy, third one is chemotherapy, then biological response modifier like vaccines, and combined modalities where surgery plus chemotherapy or surgery plus radiotherapy is prescribed or done. So I want to tell you that since the drug discovery of anti-cancer, still surgery is the best option for the removal of particular cancer or tumor from our body. Over the years, the chemist, as I said, the role of the chemist is very important. So the chemist has developed so many chemotherapeutic agents, right? We are going to focus on this chemotherapy. So chemotherapeutic agents has been developed for the treatment of cancer, right? But there are a number of problems with this treatment option. What are these problems? So number one problem is it is very difficult to target the cancer cell uniquely. As I said that there is a difference between normal cell and cancer cell, but they both are originated from our body. The only difference is their genetic makeup, right? But drug do not know the genetic makeup difference. So it's very difficult for a drug to particularly target a cancer cell and leaving the normal cell. So it's the first problem is the target. Then tumor diversity. As I said that because it's a genetic disease, so everybody has different genetic makeup. That means every tumor is unique, right? If person with X and person with Y, if both has a pancreatic cancer, it cannot be the same because both have a genetic makeup difference, both have a different DNA, right? So tumor diversity is there. That's why it's difficult to make a unique drug which can be utilized to every tumor. Number 3 is the side effect of the chemotherapy. As I said that this because of the unwanted side effect, because the normal cell and cancer cell is difficult to distinguish by a drug. That's why you will see often a side effect of the chemotherapy. And of course drug resistance, because later on once we give the more and more doses, the cell will become resistant, the cancer cell will become resistant toward the drug. And of course the limitation of animal model through which we can try to find out the efficacy of the drug. So all these parameters are difficulties which I am saying with the treatment option. Fine. Nevertheless, the researchers are working day and night for getting better and better drug for the improvement of the drug. Fine. So we'll move towards the chemotherapy. So we know that we want to cure the cancer, right? We want to target the cancer. Now see, chemotherapy is what? Chemotherapy is to kill the cancer cell, right? When you want to kill the cancer cell, there are two different ways we can kill the cancer cell. Number one, if we can target the DNA itself, the gene itself, then because DNA is responsible for cell division, so if you break the DNA or if you damage the DNA, then the DNA will become inactive, so cell cycle will stop, cell progression will stop, or another word you can say you have a tumor progression gone, right? So tumor will suppress it. So there are one approach, we call them as a targeting tumor cells DNA. There are second approach where, as I said, that the cells signaling is also important during the cell division, right? So there are number of enzymes, there are number of markers, there are number of proteins, there are number of the cell cycle regulatory genes which are not particularly functioning, normally functioning in cancer cell. So if we can target that particular cell signaling, particular enzyme, right? So by that way we can stop the cell progression, cell growth progression. So by that way we can reduce this tumor or reduce the cancer cell progression. So there are basically two types that I generally prescribe here. Okay, so for targeting tumor cell, there are a number of ways you can target the DNA. The foremost and so far the most highly utilized molecules are DNA alkylating agent. Even right now, the most of the chemotherapeutic agents are DNA alkylating agents in the clinical use. Similarly, other molecules like antimetabolite, then anti-tumor antibiotic like actinomycin and daunorubicin, then mitotic inhibitors like mitotic means the during the mitotic division there will be there is a formation of microtubules. So these Vinca alkaloids will inhibit the process of mitotic phase. Similarly, there are a number of hormones which are responsible for cancer also. So if you can target that hormones also, then also we can cure the cancer. In case of cell signaling, there are a number of enzymes which are overexpressed in the cancer cell. So we can target the tyrosine kinase enzyme, we can target the AKT enzyme or integrin link kinase, right? Similarly, tumor vascularization processes where we have a EGFR and VGFR vascularization enzymes, or cell cycle regulation like CDK enzymes, we can target that enzyme. So there are a number of enzymes which are not functioning well, either overexpressed or not expressed. There are two things, right? So overexpressed or not expressed, or you can say not working. So if we can target them, then also we can have a tremendous effect on the cell division. So the basic chemotherapy are based on these two types of division. This we need to understand. Fine, let's go ahead. So in first we are talking about the DNA alkylating agents or targeting DNA. So the chemotherapy actually started during the idea came from during the World War II. What happened is during the World War II, a sulfur gas, or you can say the mustard gas, was utilized as a war for chemical agents. So during the warfare agents, the soldiers were exposed with this mustard gas, and so many soldiers were died. And when the medical examination of the survivors, when it was done in the hospital, they found that in their blood, or you can say in their lymphocytes, they say that the number of the detail was found that the number of lymphocytes were decreased. That means the number of cells were decreased when they exposed with the sulfur mustard or mustard gas. That means the mustard gas has a cell killing ability. So this was first identified during the World War II. So later on people realized that rather than using this as a warfare gas, if we can use this kind of molecule against cancer cell, so it can kill the cell. Of course, the selectivity is a problem, but nevertheless, from 1940s to till date, about 70 or 80 years, number of agents has been developed based on the mustard, and they have a controlled toxicity. So for that, the chemist has replaced, the chemistry came into the picture, and they have replaced this sulfur atom with this nitrogen atom and with different substitution of R. You can see the wonderful change of the bioactivity, where this mustard gas, which was toxic and deadly, now it becomes a controlled toxicity of the cells, so it can use to kill the cancer cell, right? So targeting cell DNA, there are a number of molecules came into the market, number of companies are making, and right now I have just given some example here, like chlorambucin, mephalon, cyclophosphamide, temozolomide, istramustine, even cisplatin. These molecules are still prescribed right now since the discovery of 1940 to up to now. These days, molecules are prescribed. So you can say that DNA alkylating agents are the powerful molecule which are right now in the clinical use for number of cancers, right? But the problem with this kind of agent, as I said, that they are non-selective. They don't know whether this is a normal cell or a cancer cell. It can attack on both. Similarly, because you can see this, look at the structure, right? If you look at the structure, they have a very important bullet, you can say, right? Nitrogen, and it has a CH2 and halogen. Look at the whole structure, right? Except cisplatin, right? So you look at, they have a alkyl chloride, all have a alkyl chloride, right? So these alkyl chloride are very reactive. So sometimes before it reach to the DNA, before it reach to the nucleus, it will react with the other nucleophilic body and it will get quenched, right? So we'll have a less potency compared to what we expected. And of course it is a toxic, as I said, because there is no selectivity of this agent. Fine. And how to make them selective, that is what we have done some research, and we develop several molecules which are more selective. So we'll discuss later on, but these are the agents which are in clinical use. Now, how this molecule bind with the DNA? So this is the mechanism. You see that the nitrogen has a lone pair of the electron. So by nucleophilic attack, it will kick out this chlorine and it will form aziridium ion. This chemistry starts, right? So aziridium ion is a highly reactive 3-member ring system. Now this DC means deoxyguanine. Guanine, as I said, there are 4 basic units in the DNA: adenine, guanine, cytosine, thymine and uracil. So out of this, the guanine, which is having a nitrogen lone pair, which I have shown here at the bottom, this nitrogen atom has a lone pair which has a basic nature, which will attack on this aziridine ring system. It will open this ion, and now this DNA is connected with this nitrogen mustards one arm. One arm is already occupied. Similarly, this nitrogen lone pair will again kick out this chlorine and it will form again a aziridium ion, and again the second base pair. Now this second base pair can be from the same strand of the DNA, or it can be from the other DNA strand also. So sometimes it is called interstrand cross-linking or intrastrand cross-linking. So what will happen is, this nitrogen mustards alkylate the DNA base pair and it is hanging around the 2 base unit of the DNA. So that means these 2 DNA base pair are locked by this lock. Now is fixed, it cannot move. So DNA cannot replicate because it has to move, right? So when DNA cannot replicate, that means cell cannot grow. That means cell will die, right? So this how the nitrogen mustard cross-link the DNA. This is the chemistry. Fine. Similarly, we can also target, as I said previously, we discussed about the DNA. We can also target the microtubule formation. As I said that during the cell division, it's very important that cell separated by microtubules. So there are a number of plants, especially Texas, and the molecule which is isolated is wonderful molecule. This is Taxol. It's right now in the market. This molecule has been isolated from this Texas plant, and it is very useful again number of different type of cancer, especially breast cancer, ovarian cancer, lung carcinoma. But remember that the chemistry is that these molecules always target on the microtubule inhibitors, right? So another type of molecule which is vinca alkaloids. These plants is very abundant in the Asian region, especially Australia and India and even Pakistan and Malaysia. This plant produce the vinca alkaloids, especially the vincristine and vinblastine. This molecule are also microtubule inhibitors and it has been approved for number of leukemia and lymphomas and Hodgkin metastatic tumor. As I said that the DNA, microtubule, and third is inhibiting hormone, right? So chemical or molecule which can inhibit the hormone action. So there are number of hormonal related tumor is also known, right? Breast cancer is one of them, right? So estrogens and its involvement in carcinogenesis is already known since long time, right? So the estradiol and estrone, right? These kind of molecules, right, which can be utilized for the hormone related cancer, especially in case of the advanced breast cancer. The molecule like tamoxifen is approved for the anti-estrogen in 1974. So hormone related cancer, either prostate cancer or breast cancer, that can be treated by the anti-hormonal molecules. These are also synthetic molecules. So DNA targeted, microtubule targeted, and hormonal targeted. Now fourth type of targeting, we are talking about the kinases. If you look at here, I have put a picture of the macromolecules. The yellow color is a molecule which is a drug, and the other blue and gray color molecule which is a macromolecule, or you can say it's a kinase. The name of that kinase is receptor tyrosine kinase, RTK. The molecule itself is sunitinib. This molecule is useful and it has been approved for the renal cancer treatment, especially renal cancer, that is a cancer of the kidney, right? So renal cancer treatment, the molecule sunitinib is approved. Now what I'm going to show you here is that the macromolecule, especially kinases, are overacting in so many cancers, right? For example, in renal cancer, the RTK, the receptor tyrosine kinase molecule, is overexpressed. In normal cell, if you see the difference between the expression level of these kinases are very high in cancer cell compared to the normal cell. So if you can inhibit this enzyme, then also you can slow down, or you can curtail the process of the cancer cell or the progression of the cancer cell. So to stop this kinase by functioning, you have to switch off this kinase. How to switch off this kinase activity? So every kinase has a particular binding site. That binding site where natural molecule bind and kinase got activated. But here the sunitinib, which is a synthetic molecule, is known as a pyrrole and indolone combination molecule. When it bind with this tyrosine kinase receptor site, this kinase receptor site, then because this is it is not a natural ligand, so the kinase will dysfunction. Kinase cannot be activated. So the activity of kinases has been stopped. So it's like a switch off the electricity. When you switch off the light, definitely if you switch off the main switch, then definitely every light will be off. Similarly, if you switch off the one kinase, it's decreasing, or you can say the subsequent sequencing will be lost. The cell signaling will be lost. So we will have a cell killing effect, or you can say the cell static or cell progression will arrest, and cell will no more go for the division. So that's how we can target the cell by kinases activity or inhibiting the kinases. Another different type of kinases are known. Another kinases are, as I say, EGFRs, right? So endothelin growth factor receptors. There are molecules like gefitinib and lapatinib. These molecules are already in clinical use, which are inhibiting these kinases. Another type of kinases is tyrosine kinase, TKs, especially vascular endothelial growth factor receptors. These molecules is also axitinib, imatinib. These molecules are basically inhibiting the receptor tyrosine kinases or tyrosine kinase inhibitor. There are another kind of kinases is serine threonine kinases, where phosphorylation and dephosphorylation, activation and deactivation is occurring. So tyrosine kinase is different, serine threonine kinase is also different, right? So there are two type of kinases. So here, if you can inhibit the phosphorylation and dephosphorylation process of the kinases through molecule, then also you can inhibit the kinase. So there are number of serine threonine kinase inhibitors, for example, cyclin dependent kinases, which are presented here some structure. If you can see this structure, right? Similarly, PDK, then AKT. This I will talk about AKT. I have also done some research on AKT inhibitors, and I will show you that, and mTOR kinases. So these molecules are also in the clinical use for inhibiting this serine threonine kinases, right? So far what we understand is that if you want to kill the cancer cell, or if you want to cell progression, if you want to arrest the cell progression, then you have a 3 or 4 different ways. Either you attack on the DNA, or you do the enzyme inhibition, or you do the hormone inhibition. There are a number of ways we can do that. So for those people, or for learners who want to start the anti-cancer drug discovery, I have some case studies that I am presenting here, which is my research actually. So I am presenting here how we can particularly design a molecule which can be either DNA targeted, or which can be either targeting to the kinases. So first I am taking the case study of targeting DNA. So we know that the nitrogen mustards, as I showed previously, there are a number of molecules which are in the clinical use. They are very reactive, they are non-selective, because they don't know the difference between normal cell and cancer cell, and also they don't know how to reach to the cell nucleus, because they don't have any affinity towards the DNA, right? So during my research, I thought that if we can do the combination of two molecules, where we have a bullet nitrogen mustard, and if we have a DNA affinic molecule which carry this bullet to the DNA, and if we can link by some linker which is a stable linker, so we can make a more targeted. Now see, this is a DNA directed molecule, right? So we can target this, specifically target this molecule towards the DNA. So before it loses the activity, it can reach to the DNA, right? So we can make it more potent, right? So I designed some molecule, and I am presenting some of the bioactivity, not all. You can refer my research paper if you want to do further research, but here I am presenting one study case where I have used DNA affinic molecule like acridine or 9-anilinoacridine, and I have attached the nitrogen mustard with the urea linkage, and we found that this molecule were wonderfully active. So see, fine. So see that here the 9-anilinoacridine, which is a DNA affinic molecule, which is already known that this molecule has affinity for the DNA. Actually it's a DNA intercalating agent, right? So when you attach this DNA intercalating agent with the nitrogen mustard with proper linkage, you can direct this molecule, these nitrogen mustards, towards, specifically towards DNA, right? So how to attack? So the DNA interacting agents are working like this, right? So we have a linker, and we have a nitrogen mustard, and we have a director. So this director will carry the nitrogen mustard towards the DNA through a proper linker. That can be either urea or carbamate, or number of linker we can use this. This is how we design this. Now see, so how to synthesize this molecule? So we started the synthesis of nitrogen mustard. So it's very simple process, but actually it's required very cautious, because the mustard is very reactive and very toxic, right? So you start with the 4-fluoronitrobenzene, then the amination of this removal of fluorine through diethanolamine, then chlorination of this OH to chloro, then reduce this nitro to amino, we'll get a free amino. But this is a very reactive mustard, so we have to immediately make it the hydrogen chloride salt. So hydrogen chloride salt will generally stabilize this lone pair, so that you can stabilize this mustard. Otherwise it can have a lot of toxicity, and it's difficult to prepare in the laboratory without safety and precaution. And this, then later on, this mustard, I mean the nitrogen NH2 group, will be converted into isocyanate group by trifosine. This chemistry we already prescribed and it's already published. Now this isocyanate react with different anilino-acridine molecules, so we get the urea linkages. So basically we use the DNA directing agents linked with the nitrogen mustards. Fine. Look at the bioactivity of this molecule. As I said that, of course, all the molecules were very potent with the nanomolar IC50, and out of total 15 compounds, there are 2 compounds which we have studied this for in vivo study also. So look at this data. I want to just explain for a few seconds that you look at this compound 1050 and 1051, these 2 compounds that I have prepared, and you look at the data here. This blue line, which is a control where there is no drug treatment, but these 2 lines where we have a 2 drug 1050 and 1051 that we have treated. You see that after treatment start at 8 days, the tumor volume which is in control is rising, but in case of 1050 and 1051, tumor volume is shrinking in animal. Right here I saw the nude mice bearing the MX1. MX1 is a breast cancer cell. So breast cancer cells implanted in this animal, and then we started the anti-cancer therapy. So you can say that after 16 days, I mean the 5 doses treatment, right, the tumor completely gone, or you can say complete suppression of the tumor, right? And when we monitor this closely, we found that even after 70 days there was no relapse. So this was a wonderful result that ever get with the nitrogen mustard, right? So this is how the techniques of directing nitrogen mustard with the DNA affinic molecule has wonderfully worked, right? And next we try to find out that can this CH2Cl, I mean the alkyl chloride, can be replaced by something and still we can get the DNA alkylating agent. Yes. So there is one molecule known as a mitomycin C which has a capacity to alkylate the DNA by these 2 functional group OCO NH2, and this side you have NH. So it's known that mitomycin C can alkylate the DNA. So these 2 base pair DG deoxyguanine is alkylated through mitomycin C. So what we try to focus is if we can replace this part, because this part has nothing to do with the alkylation process. The very important process which are DNA alkylation, and the process involve the lone pair of the nitrogen. So what we thought that we can keep this moiety. So we, instead of doing the CH2Cl, we try to prepare this kind of bis hydroxymethyl pyrrole derivatives. So we have a nitrogen, then a pyrrole, and these 2 CH2, CH2OH or OR. So this how from nitrogen mustard, I mean alkyl chloride, to bis hydroxypyrrole we came around. See this. So this molecule, how they alkylate the DNA. So generally, like nitrogen mustard, this nitrogen will start the shedding the lone pair. So this X, which is either OH or OCO, are different functional group, can leave. So will form a Michael type acceptor. This Michael type acceptor will be alkylated by deoxyguanine, which is a base. So base will attack on this and will subsequently get back the lone pair. Similarly, this lone pair again will attack and will get the second alkylation of the same strand of the DNA or the other strand of the DNA. So we get interstrand cross-link or intrastrand cross-linking. So this how we can alkylate the DNA. So this is a second type of molecule, second type of structure, second type of precursor where we can alkylate the DNA. Instead of nitrogen mustard, we will have a hydroxymethyl. So in our laboratories with the Professor Sanlong Su, we have developed number of molecules which are hydroxymethyl, bis hydroxymethyl pyrrole scaffolds, which are potent DNA alkylating agents. And in fact, we found that they also have some other kinase type inhibition also. So just one example I am sharing here, that if we have a bis hydroxypyrrole connecting with the quinoline, isoquinoline type structure, the fuse ring system. Look at the in vivo activity of this molecule. At 30 milligram dose, you can say that this is the control where tumor is growing, high tumor volume. But in case when we treat the drug, you can say that at the day of 18 days, I mean the 6th dose or 5th dose, the tumor has shrink, right? So these also have a wonderful anti-cancer effect in animal model also. Fine. So first we will talk about talk about the how to attack the DNA. So nitrogen mustard is the first case where we can attack the DNA, or there is a second case where we use the hydroxymethyl compound, so we can attack on the DNA. Now let us talk on the kinases inhibition, how we can target the kinases. So in my research we start to use the natural compound, for example vitamin E. You know this vitamin E since a long time. It's basically known as tocopherols, or you can say the compound with double bond is a tocotrienol. So tocopherols are natural molecules, right? They are vitamin E. There are 4 types of molecule, or you can say 4 types of derivative: alpha tocopherol, beta tocopherol, gamma and delta tocopherols. So these tocopherols has a specific activity. If you know that these tocopherols are antioxidant in nature, so they remove the reactive species from our body, reactive oxygen species from our body. So during my research I try to find out that can we make this tocopherol, which is an antioxidant molecule, to anti-cancer, right? So first and foremost I thought that the lipophilic chain is sometimes a problem. So I try to cut this side chain, which is a isoprene unit. So I cut one isoprene unit, then I cut second isoprene unit. So I try to prepare the truncated isoprene side chain of these tocopherols. So how to make this? So let us discuss the chemistry. So it's about 10 step reactions where you can start from this simple acid and do the, because I want to do the isoprene unit on this carboxylic group, so I have to convert this into the aldehyde functional group, because I can do the Wittig reaction here. So for Wittig reaction I have to have aldehyde here. So from acid to ester, and from ester to reduction aldehyde, and then aldehyde to the Wittig reaction, so we get a isoprene unit, one isoprene unit. Similarly I can put 2 isoprene unit here also. So this is alpha tocopherol compound with 1 isoprene unit. Now when I want to make it to gamma tocopherol compound, I have to remove this one methyl group, because in gamma there is no methyl group here, right? So what I have done is I have to remove this methyl group selectively. So I did this through these 4 step reactions, where first I do the bromination, selective bromination here, then oxidation of this CH2Br to aldehyde function, then aldehyde oxidized to COOH, and then this COOH was decarboxylated, so I get the removal of this methyl group. So alpha and gamma both we have successfully synthesized. So you can see that you also require a strong chemistry when you want to work in the medicinal chemistry. Not only it's interdisciplinary science, but also you have to have expertise in the organic preparation, organic synthetic techniques also. So see the bioactivity of this molecule, right? We tested this compound in the lung capsule, which is a pancreatic cancer and prostate cancer cell growth. You can see this, right? So when alpha tocopherol itself is not anti-cancer, the IC50 is huge, of more than 100, not active at all. But when the side chain was truncated, when we remove the side chain and when we have a two unit, you can see that it slowly start to appearing as a anti-cancer active. And when we have only one isoprene unit on the gamma tocopherol, this molecule was wonderfully active against the prostate cancer as well as lung capsule. So when we do the animal study, we find that this compound also inhibit the tumor volume. You can see this slide here I have shown here. These tumors, these mice are produce tumor through injection, and then later on we did the treatment of the drug, and we monitor this molecule, these mice with the different days, and you can see on the right hand side of the corner that the vehicle tumor, and if you compare the compound which is treated, gamma VE5 treatment, you can see the tumor volumes are shrink, right? This is a big tumor, this is a very small tumor, so the size of the tumor is shrinking. So this is showing that this molecule is inhibiting the tumor. It's a tumor suppression agent, or you can say it's a anti-tumor agent or anti-cancer active compound, right? We try to find out whether what is the mechanism of action, and we find that these molecules are basically inhibiting the AKT. It's not targeting the DNA, right? Because there is no case that this molecule alkylate the DNA. This molecule is basically inhibiting the AKT. There is a kinase, right, which is always overacting in so many cancers. So this inhibit the action of kinase. So it inhibit the kinase via PHLPP domain. So this molecule can bind with the PHLPP domain of this kinase. So this AKT cannot phosphorylate through serine threonine kinase. So it cannot phosphorylate. So dephosphorylation will occur. So this cannot proceed for the cell progression. So and this will be published in this data, or this paper will be published in the Journal of Medicinal Chemistry. So what I want to say is that this is another way we can target the different kinases. So right now the current status of the anti-cancer drug discovery is the targeting kinases, targeting macromolecules, targeting protein, right? So that's how the anti-cancer chemistry is right now working. Now what is the future of this drug discovery? Now I want to introduce with the terminology. I don't know whether you are familiar with this terminology or not, but you heard the name of the generic drugs. So what is generic drugs? If I just want to explain just briefly, that when patent expire, that is about after 20 years, which has a license for particular molecule, right? So when patent expire, the drug become generic. That means any company can make the drug, right? So this is called generic drug. So before patent expire, they have a patent for that, so they have exclusive right. But after the patent expiry, that molecule become generic drugs, right? So generic drugs are generally small molecule. For example, paracetamol and crocin. Crocin is a brand name, but paracetamol is a generic name, right? So generic drugs, so small molecule. And when patent expire, when we make this small molecule generic drug, of course we have to make the exact molecule or exact copy of the patented molecule, and also we have to have no compromises on bioactivity. That is condition for any generic drugs. Now what is biosimilars? Now compare these two, right? Biosimilars are molecules which are either produced by bacteria or cells or living organisms. So naturally they are antibody or protein or biological products, right? So when their patent expire, you cannot make exact copy of this similar product, right? Because when bacteria or cell produce the molecule which is a protein or antibody, there will be a minor change in their structure, either three-dimensional structure or packing of the structure, or sometimes a sequence of the structure will be not exact copy, right? But their compromisation on bioactivity is not allowed, right? So biosimilar is a product where a biological product is approved by FDA when it is highly similar to the non-biological reference product, right? And that should not have any clinical meaningful differences in terms of the safety and efficacy of the reference product. That means if I talk the biosimilars, that means I am talking a protein or antibody or bioproduct which patent has been expired. So for small molecule it is known as a generic molecule, but for larger molecule or macromolecule it is known as a biosimilars, right? Because the biosimilars cannot be exactly copied, that's why minor changes is allowed, but there will be no compromises in the bioactivity. So right now since last decade, lot of biosimilar companies are interested in biosimilar product and their productions. Some of the examples that I want to share with the biosimilar products, which are macromolecules, or you can say the proteins, right, for cancer treatment, right? So alemtuzumab, which is a treatment which is helpful to treat against the leukemia. Similarly, trastuzumab, which is a active agent against breast cancer. Similarly, the anti-angiogenic cancer therapy, the bevacizumab molecule. This molecule, these all are some examples. This I have listed. There are a number of examples, but just for your information, I have listed some examples which are biosimilars in natures, and they are working as a anti-cancer agents, right? So we talk about the small molecule, and we also talk about the biosimilar products which are anti-cancer agent. But the important question always arises in the mind, that can we prevent the cancer? The answer is yes, because in natural products, or naturally, there are a number of phytochemicals are there which are our dietary sources, and which have some chemopreventive agents through which we can have a protective action, right? For example, some of the compound, or some of the figure I have used from these natural reviews, if you want, which you can go there. They have beautifully described that there are a number of phytochemicals, there are a number of phytochemicals which have a chemopreventive action, and they are dietary source. See this curcumin, right? Turmeric, which has a molecule curcumin. Similarly, chili peppers, which has a capsaicin, right? Capsaicin molecule. Similarly, ginger, which has a gingerol molecule. Grapes, this is a resveratrol, is a beautiful molecule which you can see the transphenolic compound. Similarly, honey, right? Caffeinic acid. Then garlic also has a wonderful disulfide molecule, which is a chemopreventing agents. Similarly, there are dietary sources, especially from the green vegetables. If you look at the green tea or cabbage or broccoli, the cabbage has indole 3 carbinol, which is known to have a anti-breast cancer. I mean the breast cancer molecule, anti-breast cancer molecule. Similarly, the broccoli has a sulforan molecule. Then soybean also have a genistein molecule. Similarly, tomatoes has a lycopene. So all these are a dietary source molecule. They contain the molecule, or the chemistry is beautiful, that it can work as a chemopreventive agent. So yes, it is possible for certain extent to prevent the cancer. So before I conclude, just I want to review the entire presentation. What I have discussed today is, we talk about the entire drug discovery for the anti-cancer. We talk about the brief introduction of the cancer, different treatment options. We talk about the hallmarks of the cancer, and I have presented some case studies of my own research, where we can target the DNA, or we can target the enzyme also, and we can wonderfully contribute into the anti-cancer drug discovery. And I want to acknowledge some of my very own institutes where I have worked. Institute of Biomedical Sciences, Academia Sinica, Taiwan, and the Ohio State University, the College of Pharmacy, where I have done my postdoctoral research. And presently, I want to thank the Department of Chemistry and Forensic Science, Bhakti Kavi Narasimha University, for their patronage. Thank you all.